This is podcast 14. Okay, so we're going to get into some metabolic pathways. As an introduction to those metabolic pathways, how you break down carbohydrate, how you break down protein, how you break down fat. We'll talk about the end goal for a lot of these pathways, which is ATP. And ATP is a common energy molecule. It's not the only one. There's also things like GTP and creatine phosphate. But the idea is that you have a molecule with potential energy that's easily accessible, and you can release that potential energy and use the energy to do work. The work can be synthetic work, where you're putting together monomers to make a dimer and a trimer and a polymer. Can be concentration work, so let's say you're acidifying a vesicle in the Golgi apparatus and you need to pump it full of hydrogens. That's concentration work. It could be electrical work in that you're moving ions so that you can create an electrical gradient in, say, a neuron. It can be mechanical work, such as you saw with those motor proteins walking along a microtubule. Could be bioluminescent work. We haven't talked about this specifically, but one of the products of these reactions can be light, and organisms can glow as a result of that. It can also be wasted energy that is released as heat from a chemical reaction. You don't harvest all of that potential energy into work. You're going to waste some as heat, and this is where most people think your high internal body temperature comes from, is from wasted heat from breaking ATP and breaking other bonds. In order to synthesize ATP, you've got to have a free energy that is high enough so that you can actually put a phosphate onto ATP, and we'll talk about how that happens. So ATP as a molecule is should look really familiar to you in terms of the macromolecules. It looks just like a nucleic acid. Essentially, it is. You have a base and a sugar and a nitrogenous base, and then 3 phosphate groups attached off of one of the carbons off the sugar. If it's 3, then it's adenosine triphosphate. If it's 2, diphosphate. And it can also be 1, AMP, not shown on this diagram, and then a free phosphate that is released as PI. So why is ATP the go-to molecule? Well, it's right in the sweet spot for providing a large amount of potential energy, and it's a largely exergonic reaction. So you remember from other classes that a negative delta G means that the reaction will be spontaneous, and the magnitude of the negativeness is an indicator of how spontaneous is that reaction and how much energy is released. So when ATP is split into ADP plus P, releases 7.3 kilocalories per mole. And this is always often talked about a high energy bond that is being released, and that's a little bit misleading. What the details of that mean is that the amount of energy that you need in order to break off that third phosphate is less than the amount of energy that is released as a result of breaking off that third phosphate. So you just need a little bit to knock off that third phosphate, and you release a lot of potential energy when you do that. And why do you release a lot of potential energy? Well, these bonds, these phosphoryl anhydride bonds, you're holding up together this largely negative thing against to another thing that is largely negative. And so there's charge repulsion, like trying to hold two magnets with opposing poles together. And when you release them, there's a large amount of potential energy released. Another idea is that this structure here, as a free phosphate, distributes the electrons evenly across all four of the oxygens. So it's a small case Greek delta, means partial negative charge across all of those. But in this structure, it's not evenly distributed. So in intact ATP, there's more potential energy because of a differential charge distribution that gets released to a more equal charge distribution when you release that free phosphate. So this is another source of energy, potential energy that gets released. And then releasing the free phosphate increases entropy, the amount of disorder in the system. Now you have a phosphate that is not constrained in a bond to another phosphate. It's a phosphate that can be free and then has less ordered less order to the system, more entropy in the system, and increasing entropy is increasing negative delta G, and that drives the reaction toward this direction, which is release of the free phosphate. Okay, so I said it's not particularly a high energy bond. So let's say here's a bunch of spontaneous reactions, and the lower the number of delta G, the more spontaneous it is. So this is a very spontaneous reaction. You're releasing 14.8 kilocalories per mole when this reaction happens. This is not a very spontaneous reaction, only releasing 2.2 kilocalories per mole. And ATP is someplace in the middle. So using shorthand in another class, when instructor says this is a very high energy bond and this is why we use ATP, it's pretty middle of the road as far as how much potential energy is in there. But that's also an advantage as to why ATP would be good. Because if you want to move phosphates around, you can only move them from a higher energy compound to a lower energy compound. So for example, if this phospholinopyruvate is catalyzed to pyruvate plus PI and releasing that phosphate could be added on to ATP, for example, that has a lower delta G, because it does have a lower delta G. Or ATP could release its phosphate to become ADP plus P, and that phosphate can be added on to glucose 1-phosphate to become glucose 6-phosphate. That could be added on to glucose to become glucose 6-phosphate because this has a lower delta G than ATP does. But if I want to go the other way, take the phosphate off of glucose 6-phosphate and add it on to ATP, can't do that because I have to add energy in order to do that. It would not be spontaneous. The reactants would not have less energy, or the products would not have less energy than the reactant, so it wouldn't be spontaneous. So being middle of the road, you can do some of these reactions, which are indeed reactions that happen in glycolysis. And if it was at the top of the curve, you might not have as much flexibility. Okay. Another thing we're going to run into in glycolysis is the electron carriers. And these are, again, very nucleic acid-like in their structure, not as obvious as is ATP or GTP, but they look similar in that they have a sugar and then a base. In the case of NAD, it can accept an electron and become reduced in the form of this extra hydrogen here on this ring. And this reduced molecule is a way of storing that energy of that electron. So reduction is adding on of electrons. Oxidation is stripping away of electrons. So when these compounds are reduced, they're storing that electron's energy that happens in these intermediate metabolism pathways, and then typically toward the end of the pathway, you take these electrons off and you pass them down an electron transport chain to reap the energy back. So it's a way of transferring energy from one part of the reaction to another part of the reaction, and you'll see this in NAD becoming NADH when it becomes reduced, and then back to NAD when it gets oxidized, and you'll see it also in another one called FADH2. These are related or have components to them of the B vitamins. So you may have heard about people taking a B vitamin shot or a B vitamin supplement for energy. So most of that is hooey, in that you should get plenty of this in your diet, but at least there's some idea there that it's related to energy. If you did have a deficiency of NAD, you would be compromised in your ability to make ATP, and you'll see why in a second. Okay, here's an H5P question for you. Okay. Getting ready to go over glycolysis, what's going to happen is that glucose is going to get split into two, glucose is a 6-carbon molecule. It's going to get split into two 3-carbon molecules. And then those 3-carbon molecules are going to take the phosphates from them after they've been rearranged a bit, and then move those phosphates onto ATP. And then it happens again at the end of the reaction. So first, what's going to happen is you're going to spend some ATP to activate this glucose molecule and split it in two. Those molecules are going to get rearranged a bunch. They'll take phosphates off of substrates that get phosphorylated during the reaction and add them onto ATP at two different spots. So you'll spend 2 ATPs, make 4 ATPs for a 2 net gain. This kind of making ATP is called substrate level phosphorylation, because the phosphate is taken off of a substrate and added onto ATP. Later on in the next podcast, we'll talk about oxidative phosphorylation, where the energy from oxidizing those electron carriers I just talked about, NAD and FADH2, is used to take free phosphate and add it on to ADP, and that's called oxidative phosphorylation. It's called substrate level phosphorylation because the phosphate is taken from a substrate, a carbohydrate 3-carbon molecule, and added on to ATP. Okay, so here's the big old pathway. I'm going to walk through this. And this is glycolysis, sometimes called stage 1. This happens in almost all organisms. It typically happens in the cytoplasm. There's some parasites where it happens in a membrane-bound compartment, but most most of the time happens in the cytoplasm. You've already seen this in some form or another, at least in one other class, probably more like 2 or 3 other classes. Why do we talk so much about glycolysis? It's because it's one of the best known pathways. It's one of the oldest known pathways in that most organisms on the planet have it. Also oldest to science, one of the first things that had been worked out, what transfers to what, and it's still relevant. So glycolysis is important in cancer treatment. Glycolysis is important in omics sorts of experiments where you're trying to figure out what proteins go up or down. And these proteins that catalyze this reaction are some of the most abundant in your cell. So for all those reasons, we're going to start off with glucose, which is a 6-carbon molecule. Glucose, the first enzyme, all these arrows are enzymes. The first enzyme, GLY1, is called hexokinase, and it's called hexokinase because it works on more than one hexose, or more than one 6-carbon sugar. So glucose is not the only thing it works on. So the reaction is catalyzed to add a phosphate onto the number 6 carbon in the glucose. 1, 2, 3, 4, 5, 6. Number 6 gets a phosphate added onto it. That phosphate comes from ATP. So we're supposed to be making ATP, but here we're spending ATP. You can sort of think of this as activation energy, or you got to spend money to make money. Okay, so now this glucose has a phosphate on carbon number 6. Enzyme number 2, which is called phosphoglucoisomerase, will change this glucose molecule into another 6-carbon molecule that has a different arrangement of its carbons. So if you notice, the C double bond OH in glucose 6-phosphate is now that C double bond O is on the number 2 carbon instead, and this is now called fructose 6-phosphate. So it's another 6-carbon sugar that has been rearranged into a different type of carbohydrate, still 6-carbon sugar, and still with a phosphate on carbon number 6. Glycolytic enzyme number 3, phosphofructokinase 1. Okay, there's another one that does a different sort of thing someplace else, and that's why it has a 1 next to it to distinguish 1 from 2. But this is 1, PFK1, catalyzes the addition of a second phosphate onto this fructose 6-phosphate onto the number 1 carbon. Number 1, 2, 3, 4, 5, 6. Carbon number 1, carbon number 6. Fructose 1,6-bis means 2 phosphates. So phosphate on carbon number 1 and carbon number 6, and there's 2 of them. Okay? Then glycolytic enzyme number 4, aldolase, catalyzes the reaction to make dihydroxyacetone phosphate and or glyceraldehyde 3-phosphate. So these 2 molecules are both possibilities coming out of aldolase. And the enzyme triose phosphate isomerase, glycolytic enzyme 5, can interconvert these easily. So you get either this or this. This glyceraldehyde 3-phosphate is the one that's going to go forward. So if you make this, you're going to convert it into this anyway. And there's 2 of these. Okay, so notice these are 3-carbon molecules, 1, 2, 3, 1, 2, 3. So if you make 1 of each, this one gets converted to this one. If you make 2 of these go forward for every glucose that you started with over here. Okay, so let's just assume we're going forward with glyceraldehyde 3-phosphate. It's phosphate on carbon number 3. Glycolytic enzyme number 6, glyceraldehyde 3-phosphate dehydrogenase. It's going to do what it says. It's going to dehydrogenate this glyceraldehyde 3-phosphate, and it's going to grab a phosphate from the cytoplasm. you're constantly splitting ATP, and there's going to be a lot of free phosphate in the cytoplasm, so there's phosphate around, and takes that phosphate from the cytoplasm and adds it onto the number 1 carbon. At the same time, NAD plus gets reduced to NADH. So in principles, I talk about charging up a battery. These are electron carriers, just like batteries charge up and carry electrons. This NADH, you're going to see in the next podcast how you're going to get that energy back. But this is a way of storing energy from breaking down glucose. Glucose is being oxidized as it goes through the process, and you're getting some of that energy back right there. You're just not going to get to use it right away. So now you have 1,3-bis phosphoglycerate. So carbon number 1, carbon number 3 has a phosphate on it. There's 2 phosphates, so it's bisphosphate, and the molecule is glycerate. Okay, and there's 2 of these molecules per 1 glucose starting. 1,3-bisphosphoglycerate, glycolytic enzyme number 7, phosphoglycerol kinase, takes ADP and takes one of the phosphates off of 1,3-bisphosphoglycerate and adds it on to ATP. Okay, and because there's 2 of these molecules, you make 2 ATPs, and you end up with 3-phosphoglycerate. So it used to be 1,3-bisphosphoglycerate. The number 1 carbon got taken off. You ended up with 3-phosphoglycerate, and phosphoglycerol kinase does that. Kinases are enzymes that add phosphates. In this case, the phosphate gets added on to ATP. So because it's taken off of this substrate, 1,3-bisphosphoglycerate, and added on to ATP, this is called substrate level phosphorylation. It's very different than how we're going to make ATP in the next podcast. Okay, so I've made 2 ATPs here. I spent 2 ATPs here and here, 1 here, 1 here. So at this point, I'm even Steven from where I started, except I have 2 NADHs from doing this reaction twice. Okay? 3-phosphoglycerate, glycolytic enzyme number 8, phosphoglycerol mutase, changes that to 2-phosphoglycerate, just moves the phosphate from the 3 carbon to the 2 carbon. Enzyme number 9, enolase, in glycolysis converts 2-phosphoglycerate to phosphoenol pyruvate, rearranging this molecule, turning into phosphoenol pyruvate from 2-phosphoglycerate. Okay, and in the last step, pyruvate kinase, glycolytic enzyme 10, it's going to take the phosphate off of phosphoenol pyruvate, add it on to ATP. Again, it's a kinase, so it's going to add phosphates onto something, in this case ATP. And because it's taking it off of this substrate, that phosphate right there, and adding it onto ADP, it's called substrate level phosphorylation again. And the end product is pyruvate, a 3-carbon molecule. You have 2 of these per the 1 glucose that you started with. And that's the end of glycolysis, also called stage 1. So, this is to remind me to tell you that thinking about these biochemical pathways, I think it's useful to think of a giant pipeworks, and the flow of material is through all these pipes, okay? And that can be hard to determine how much flow is happening through the pipes. So you can just go to the outlet, let's say it's down here someplace, and just measure how much is coming out the other end. But if you want to know at what point in this giant pipeworks is the rate at which it's coming out determined at which point in this pipeworks. At some point in this huge thing, there's going to be a restriction in that's going to be the slowest flow through that one point. And your rate at which it comes out at the end, when you're all done, is determined by the slowest point in the pathway, and that, in the case of biochemistry, is called the rate-limiting enzyme. In the case of glycolysis, it is the third enzyme, phosphofructokinase 1, which is the rate-limiting enzyme. So, if you want to know the capacity for glycolysis in an organism, you can look at the rate at which phosphofructokinase operates. What is the rate at which it can catalyze a reaction? Because it's the slowest in this pathway. If you know how fast that can go, you know how fast glycolysis can go. You can get a more nuanced idea by measuring all these enzymes, and you can have a much better idea, well, not much better, but you can have a more refined idea of the flux that's happening at every one step. But that's a lot of work, and if you just measure what is happening at the rate-limiting step, you can get an idea of how much capacity that cell has for glycolysis. This is a really common tool in the lab when scientists or clinicians want to know what the capacity is for a particular pathway. They will look at the enzyme that is the rate-limiting step. And so for glycolysis, that would be PFK1. Okay, here's an H5P question for you. Okay, these sorts of podcast lectures always make students nervous because there's a lot of detail and you always want to know, what do I got to know? So, what do you got to know? You got to know names of all the reactants, all the products, and all the enzymes. You got to know all those names. You should know the order of the reaction, what comes first, what comes next. How oxidized is something relative to something else? So glucose gets oxidized as it goes from starting from glucose all the way to pyruvate. Each step along the way gets more and more oxidized. So if you compare any two steps, you should be able to tell which one is more oxidized relative to the other, depending on, is it closer to the glucose side or is it closer to the pyruvate side? And you should know the number of carbons in each intermediate. Okay?